Okay, so welcome everyone to the last session. It's my pleasure to introduce our last but not least invited speaker, Mary Alberic Jorda, who is the head of the Laboratory of Hemato-Oncology from the Institute of Molecular Genetics of the Czech Academy of Sciences, and she's gonna give us a talk on hematopoiesis. So please take it away.
Thank you, Martin. So welcome everybody to this last session of this meeting. First of all, I have to congratulate Martin for this awesome program that you have set up together. I have to say it was very diverse and at the same time complementary. So my congrats. And also I want to thank you for giving me the opportunity to present some of the work that we are doing in the Laboratory of Hemato-Oncology. You will see throughout my slides that I have this upper left corner empty with a square. You can, if you want, drag your Zoom window so it's not going to be all over the slides. So to start with, let me introduce you the granulocyte. So a granulocyte is a blood cell type, maybe you already know it, but today I will try to convince you that these cells and the production of these cells is very important for our organism. So what a granulocyte is doing is representing the first line of defense in our organism when we are facing a pathogen. Let's say we get challenged by a bacterial or fungal pathogen, and the granulocyte will be the first cell type that will go to the site of infection to try to clear up that pathogen. So the granulocytes do have certain properties that make them unique and very efficient in their task. So they can phagocytose, they can secrete granules, and do natosis, for instance, and all these activities are aimed at killing the pathogen present in the organism. They can also activate a pro-inflammatory response, and that means that they are going to create some signals based on the secretion of cytokines that will call additional immune cells to come to the site of infection and help clearing out and fighting this infection. And all this coordinated response aims at preventing what we know as a systemic infection. So now that we know what the granulocytes are doing, let me tell you how do we produce them. So in healthy conditions, our granulocytes are being produced in the bone marrow. This is what we know as a steady state granulopoiesis. So the bone marrow, hematopoietic stem and progenitor cells do give rise to these granulocytes. And once they are mature, they can exit and move into circulation. They will be there for around 3 to 5 days and then they are gonna die. Therefore, the bone marrow needs to constantly replenish and produce new granulocytes. When we are facing a pathogen, we need to activate what we know as an emergency granulopoiesis program. And this is a response that is meant to, first of all, activate the proliferation of our stem and progenitor cells, and second, induce huge and large numbers of granulocytes in order to fight this infection. Once the infection is cleared out and the pathogen is not present anymore, we need to go back to the steady state conditions. And this controlled production of granulocytes, adjusting itself at the different conditions we are facing, is very important in order to keep our organism healthy. What happens if the production of granulocytes is not properly regulated? If we produce too few granulocytes, we might develop neutropenia. Patients that do suffer from neutropenia do have recurrent infections that can lead to lethal conditions. If we produce too many granulocytes, we can develop autoinflammatory disorders. And these autoinflammatory disorders, I'm sure you're familiar with, for instance, diabetes, rheumatitis, and arthritis. Excess of granulocytes is also not good, for instance, in COVID-19. Someone told me, if you want to get the attention of the audience nowadays, you need to have the word COVID-19 in your talk. So here it is, my COVID-19 slide. Granulocytes are also very, they have a very negative impact in the flow of the disease. So they can contribute to what is known as systemic inflammatory syndrome, which is what most of the patients in the intensive care unit are suffering from. If the production of granulocytes is not properly occurring, we can also develop leukemias, for instance, acute myeloid leukemia. What we have is the hematopoietic stem and progenitor tool that can only partially differentiate. The granulocytic differentiation gets arrested at certain point, and that leads to the accumulation of immature and non-functional blast cells, leading to leukemia. So if we look at the hematopoietic tree, we see that at the top of this hematopoietic hierarchy, there is the stem cell. It can give rise to myeloid progenitors as well as lymphoid progenitors. And then further proliferation and differentiation of those cells will give rise to all mature cells we encounter in blood, including the granulocytes. We have seen already during these days that the hematopoietic stem cell needs to maintain this self-renewal potential, and it has a very low degree of differentiation. As the stem cell loses the self-renewal capacity, it gains in differentiation. So there is an inverse correlation between these two properties of the hematopoietic stem cell. Now, we have also learned that new technologies like single-cell RNA sequencing have challenged the view of this hematopoietic tree. We still believe that at the top of the hematopoietic system, in the center, there is a hematopoietic stem cell. This one has still the ability to give rise to all mature cell types, and it commits to the different lineages. However, we do not face this linear progression anymore. We think that there is a cloud or a continuum of differentiation where the progenitor cells retain this plasticity or ability to give rise to any mature blood cell in many, many different stages, almost at the later stages of differentiation. So in my laboratory, what we are trying to do is to understand the molecular mechanisms that regulate granulocytic production. And for today, I would like to present to you a project where we have been studying the role of the Wnt signaling pathway in hematopoiesis. And why did I choose this project? The reason is that there is plenty of papers out there investigating the role of the Wnt signaling pathway in hematopoiesis that have presented contradictory results. And I'm talking about journals like Nature, Science, Cells Stem Cell, like very good journals with very solid data, but the papers conclude opposite results. So it's very difficult to understand once you enter literature what is really the role of the Wnt signaling pathway in hematopoiesis. We think that all this variety of observations is due to the fact that this pathway is extremely complicated and it has many players. So many experimental models have been designed where they modulate expression or overexpression, knocking out. All models exist out there of different proteins. And what's happening is that depending on the protein you are playing with, the results might be different. It also, there is another component that is the fact of modifying the beta-catenin signaling pathway, this Wnt signaling pathway, in a stem cell versus a progenitor cell. That might also lead to different results. Also, using murine cells versus human cells might have different output. And of course, if you study this in the context of healthy environment or a leukemic context, the role of this pathway can be different. So in my lab, we try to bring some light into this pathway, into this knowledge, or maybe some more confusion, I'm not sure. But we have generated a new mouse model that will try to address the canonical Wnt signaling pathway in hematopoiesis. So before I present to you our model, I have to say a little bit about Wnt signaling. Wnt molecules are secreted and present in the outside of the cell. When those ones are absent, the key player of this pathway, beta-catenin, gets phosphorylated by GSK3 beta. When beta-catenin is phosphorylated, it's target to degradation. In the nucleus, what's happening in this context? So we have the TCF-left transcription factors that are the ones in charge of mediating this Wnt signaling bound to Gaucho. What happens when you have Gaucho TCF-left is that the transcriptional complex is inactive. And although it is bound to these Wnt recognition elements in the DNA, the target genes cannot be transactivated. On the contrary, when you have Wnt present in the environment, the Wnt ligands will bind to its receptor, which is Freesel. Freesel receptors will then recruit axin and GSK3 beta, disassembling this phosphorylation complex. So now beta-catenin cannot be phosphorylated anymore. What happens when it's not phosphorylated is that it's not target to degradation anymore. And in this way, beta-catenin accumulates in the cytoplasm. Once it accumulates there, it's able to translocate into the nucleus and bind to a specific site of the TCF factors, and therefore displacing Gaucho and allowing transactivation of the Wnt target genes. So to investigate this pathway, what we did is we generate a mouse model that would specifically inhibit the beta-catenin TCF-left binding. And how we did that is by introducing a dominant negative form of TCF4. This dominant negative form of TCF4 does not allow beta-catenin to bind to this transcriptional complex. So then Gaucho cannot be displaced. And as a consequence, even beta-catenin is accumulated and present in the nucleus, it cannot lead to transactivation of the Wnt target genes. Now, if this was a little bit complicated, I just want you to just retain this idea in mind. It's an easier view of the model. So in the wild-type mice, we do have active beta-catenin TCF-left mediated transcription, but in the DN-TCF4 mice, this transcription is inhibited, is impaired. So in my lab, one of the techniques we use very often is flow cytometry. So what we do is we take wild type, and in this case, DN-TCF4 mice, isolate the bone marrow and blood, and put them through FACS analysis. We have seen many plots these days out here already, but I just want to bring out little cartoon of what we are doing. So this is a whole bone marrow, and we stain it with cell surface markers. And by combining expression of these cell surface markers, we can identify several populations in the bone marrow, such as progenitor cells, stem cells, and granulocytes. And throughout my presentation, I will be talking sometimes about these c-KIT positive cells, which are myeloid precursors, myeloid progenitors, and LSK cells, which combine mainly progenitors and stem cells. If we look at the bone marrow of wild type and DN-TCF4 mice, and we perform flow cytometry, we get something like this, which is quantified below. You don't have to retain all the information, but the idea in here is that the DN-TCF4 mice, shown here by these red bars, do have increased numbers of stem and progenitor cells. It seems that they have been accumulating. We next look at the blood of those animals, and I will focus on the granulocytic population. So by using LYCG and LY6C, we can identify a population of double positive cells, which are our granulocytes. And you can see in these FACS plots, as well as in the quantification next to it, that the percentage of granulocytes was reduced in the blood of those animals. Now, to better understand what was happening with the bone marrow of the DN-TCF4 animals, we perform serial replating assays. So the way that this works is we take a bone marrow and we put the cells in culture. The stem and progenitor cells will have the ability to form colony-forming units. The idea is that each of these units or each of these colonies has been derived from one stem progenitor cell. As healthy bone marrow cells, they will not be able to be replated forever. And that's what you see over here. You have the first plating, nice number of cells. After 7 to 10 days, you can replate them. They are still here, but then they lose this property. If you look at the cells, the morphology of those cells in the second plating, you can see that the cells are showing a granulocytic-like shape. And also, the cells do express high levels of mature granulocytic markers. So this is what's happening when you put wild-type cells in culture. Now, if we look at the DN-TCF4 bone marrow cells, what's happening is that they have an enhanced replating ability. They do show an immature-like morphology, as you can see in the pictures over here, and also expression of mature neutrophilic markers like CBP epsilon and lysozyme are reduced. On the contrary, the expression of immature markers like neutrophil elastase and cathepsin G have been increased. So altogether, this brings the idea that the beta-catenin TCF-left signaling pathway is required to induce granulocytic differentiation. But of course, our question is how? How does this beta-catenin TCF-left play a role in this process? And actually, when we don't have a clue about how to address this question, what we do nowadays is RNA sequencing. So we just take bone marrow from wild type and DN-TCF4 animals, isolate the hematopoietic stem and progenitor cells, and we perform RNA sequencing to have an idea about the gene expression profile of those populations, of those cells. We had 3 DN-TCF4 samples and 3 wild-type samples. And when we perform unsupervised clustering, and you can see that on this heat map, the DN-TCF4 clustered in one plate and the wild-types in another one. And also, by looking at the colors, we could see that some genes were upregulated and downregulated in the DN-TCF4 mice in comparison to the wild-type. Another way to show this data is by looking at the volcano plot. In here, we can see that about 700 genes were downregulated and about 700 genes were upregulated in the DN-TCF4 stem and progenitor cells of the DN-TCF4 mice compared to the wild types. And when we took a closer look into these downregulated genes, we saw that there was an enrichment of genes that are important to orchestrate granulopoiesis. For today's talk, I would like to focus on GCSF receptor. This is too small to really get into it, but the idea is that GCSF receptor recognizes GCSF, or GCSF binds to GCSF receptor, activates the signaling cascade, which includes phosphorylation of STAT3. Phosphorylation of STAT3 will activate transcription of genes required for granulocytic differentiation. And what we have seen and then also verified was that GCSF receptor was downregulated in DN-TCF4 stem and progenitor cells compared to the wild types. And we also quantified GCSF receptor protein levels, and we observed that in stem and progenitor cells, that was also downregulated. Then we took those cells and we simulate them with GCSF and analyze phospho-STAT3 levels at different time points. When we check phosphostat-3 levels in wild-type cells, we could nicely see an activation of the signaling here. However, when the experiment was performed using DN-TCF4 cells, we saw that activation of phosphostat-3 was reduced, indicating that the response to G-CSF is diminished, probably due to the reduction on G-CSF receptors. So this brings us to the second conclusion, where we can state that the beta-catenin TCF-mediated transcription imposes expression of genes that orchestrate granulopoiesis, for instance, G-CSF receptor. I've told you at the beginning that granulocytes are important to fight an infection. And we know that G-CSF receptor is a key player of emergency granulopoiesis. So, of course, the next logical question for us was to investigate what was the role of this pathway in the context of infection. And to do that, we induce emergency granulopoiesis in our mice. The way we do that is by injecting LPS, lipopolysaccharide, into the mice, and that mimics a bacterial infection, systemic, and after a while, we analyze those animals. What you would expect is that, first of all, there is mobilization of granulocytes from the bone marrow to the blood, and then you would also expect that in the bone marrow, there is an increased number of myeloid progenitors. When we look at peripheral blood of mice challenged with LPS, we could see that the DN-TCF4s, shown here in red, had reduced number of granulocytes in blood compared to the control animals that were also injected with LPS. And when we took a look to the bone marrow progenitor cells, one of the hallmarks of emergency granulopoiesis is expansion of the GMP population, which stands for granulocytic monocytic progenitors. However, this expansion that was nicely seen in the wild-type mice was not so visible, or actually was neglected in the DN-TCF4 mice. But was this relevant at all for the animals? Had any biological implications? And we perform a survival experiment. Here we inject animals with Candida albicans, and we assess the ability to fight and survive infection. And what we could see is that the DN-TCF4 mice were succumbing to infection more often than the wild-type animals. And of course, why was that happening? We had a couple of options to explain that phenotype. And one of them, the first option, would be that the granulocytes from the DN-TCF4 mice were non-functional. So what we did is we isolate granulocytes from wild-type and DN-TCF4 mouse and perform some assays where granulocytes play a very important role. For instance, we did ROS production, we checked migration, and we also analyzed phagocytosis. However, wild-type and DN-TCF4 mice exhibit the same ability to produce those effects. So we believe that the function of the DN-TCF4 mice granulocytes was not affected, and that could not explain why the animals would succumb to Candida albicans infection. Our second option was that what's happening is that the bone marrow of the DN-TCF4 mice, in particular the hematopoietic stem and progenitor population, would fail to fight stress. So the way to assess that was to challenge our mice with 5-FU, this 5-fluorouracil. What this drug is doing is wiping out all mature cells that we have in the bone marrow. Actually, it's frequently used as a chemotherapy in the hospital. So We subjected the animals to serial injections of 5-FU to challenge the hematopoietic compartment, but in particular to challenge the stem cells. If you look at the middle panel, you can see that 7 days after the first dose of injection, granulocytes were ablated completely in wild type and the NTCF4 mice. However, 14 days later, the wild type animals had the ability to rescue that absence and start producing large number of granulocytes, but that was not visible in the DNTCF4 animals. And actually, when we gave serial injections of 5-FU and assessed how animals would be behaving, we observed that DNTCF4s would succumb to the injection while the wild-type mice will still be alive, indicating that this hematopoietic stress at the stem cell level was what was really causing the animals to die upon infection. So the third conclusion today would be that the beta-catenin TCF4 left transcriptional complex is required for proper emergency granulopoiesis. Now, once we have done all this work with mice, which we think it was a good model to get into the mechanistic role of this pathway in hematopoiesis, we want to know whether this is any relevant for human granulopoiesis. And then what we did next is we isolated CD34 positive cells, which are stem progenitor cells, from the human cord blood, umbilical cord blood. Those cells were isolated and infected with retroviral particles that would induce overexpression of DNTCF4, like we had in our animals, or we also infected them with an empty vector controlled retroviral particle. When cells were infected, they would activate our GFP reporter, and that would allow us to sort the cells and assess them in vitro. So we first expand them, and then we put them into a granulocytic differentiation cocktail and analyze them by FOX. So we took 3 different human granulocytic markers. Those are CD11B, CD18, and CD66B. And we assess expression of those markers in cells that were infected by DNTCF4 or empty vector. Here we have 2 samples, 2 human samples that were analyzed. And you can see that cells infected with MSCV, shown here in green, had higher expression of those markers in comparison to cells that were expressing DNTCF4, indicating that introduction of this DNTCF4 would impair granulocytic differentiation of human primary cells. Now, if you think about bringing some of our observations into clinics, you might think about using some drugs. And for that, we focus on cercosporin. So cercosporin is a small chemical inhibitor that has been shown to prevent the binding of beta-catenin to TCF4. So it exactly mimics what DNTCF4 is doing. So in this case, we took the human CD34 positive cells, we expand them for 5 days, and then we put them in differentiation media, differentiation conditions, in the presence or absence of cercosporin. After that, we perform flow cytometry and assess expression of granulocytic markers such as CD66B, CD15, CD16, and CD11B. And what you could see is that while presence of, let's say, diazo control would allow cells to differentiate towards mature granulocytes, the introduction of cercosporin into the cultures would reduce that differentiation ability. So what we see with this experiment is that our genetic model can be mimicked by a drug or a chemical inhibitor. And this would bring me to our proposed model, So when the beta-catenin TCF4 transcriptional complex is active, it induces expression of GCSF receptor. And expression of GCSF receptor in hematopoietic stem and progenitor cells will allow them to detect GCSF and produce granulocytes. In the context of emergency granulopoiesis, we would be able to expand this granulocytic pool. However, when we inactivate the beta-catenin TCF-left signaling pathway, now beta-catenin cannot bind to the TCF-left factors and it cannot induce transactivation of target genes, such as GCSF receptor. Now, the levels of GCSF receptor are reduced in this compartment, and consequently, the production of granulocytes is reduced. In addition, in the context of emergency granulopoiesis, where there needs to be a huge expansion of the granulocytic compartment, this cannot occur when the beta-catenin TCF4 transcriptional complex is inactivated. And this would bring me to my acknowledgement slides. First of all, I want to thank people in my lab, especially Peter Daniel, that was the driving force of this project. I want to thank our collaborators at IMG, like Vladimir Korinek and Tomasz Brewiczka's group. Our collaborators at the second faculty of medicine here in Prague, so Sharka Rakhmatova and Robbie Pitlik. We also want to thank our collaborators in Singapore and our collaborator at Harvard Medical School in Boston. And of course, I want to thank you for your last piece of energy during this conference, and I will be happy to take any questions.
Thank you. So again, please use the participants and raise hand function should you have any questions to Mary. So I would like to ask a question, and this is sort of I'm not sure how exactly relevant it is, but you tell me. So for instance, under pathological conditions, so in the bone marrow, there are immature myeloid cells that are produced, and under pathological conditions, when they are activated, they differentiate into suppressive phenotype, like the granulocytic myeloid-derived suppressor cells that Elina was talking about in her talk. And so I wonder, of course, these cells play a role in a number of pathological conditions, including cancer dissemination, of course, and but they've been discussed in pregnancy and other conditions and all that. And so, of course, one of the possible therapeutic approach is to sort of force the differentiation of these GMDSCs into their final differentiation state, so namely just your regular friendly neighbor neutrophil or macrophage in terms of when it comes to monocytic. So I wonder whether the cercosporin drug, whether this can sort of induce a differentiation, or whether this is just I'm not sure whether this might be just asking you for a wild speculation.
Sure. So we did actually try several Wnt pathway activators and inhibitors. And with cercosporin, we inhibit the pathway and we block differentiation. But we did use a couple of activatory drugs. They were not based on direct activity at the transcriptional complex, so they work at the upstream level of the pathway, but we saw that by activating the pathway, we would be pushing the granulocytic differentiation. So actually the idea would be that by modulating the pathway using different drugs, we could be adjusting the granulocytic production to our clinical needs.
Yeah, so I'm also asking because you mentioned that the receptor for the granulocytic colony stimulating factor. I always get the shortcut wrong, but anyway, so it's downregulated in the DNTCF4 mice model. But so I wonder, because I think the myeloid-derived suppressor cells are actually responsive to. Might be mistaken here, but I think they do respond. So they might, require a different approach because of the different effect that you observe in this case. So there's a difference between your case and then what's going on with the pathologically activated cells.
I mean, I do not have an answer to that question because we did not study inhibition of this complex in pathological conditions. So, yeah, I mean, as I said at the beginning, many articles did give rise to contradictory results. And I do not think that the conclusions that we are reaching are wrong. I just think that it depends on the context. So as you are saying, in some clinical pathologies, the role of this pathway could be totally different, and activation could be leading to activation or production of granulocytes in a different manner, or suppressor cells, myeloid suppressor cells could be different.
Thank you. So, time for other questions. In that case, I will ask another. And this is precisely just to my own philosophical interest here. So I'm wondering. So you mentioned that you sort of proposed a model, a mechanistic model, which you expressed in the form of a diagram. Maybe could you share again the slide, please? Yes. That would be helpful. Yeah, this is it. Thank you. So I don't see it anymore.
I think it takes a couple of seconds to go up.
Okay, but in any case, the question is extremely simple actually. So this is the end product of your research, right? So it's the late stage of the experiments that you were describing before. So including the development of the new mouse model that allows you to perform the studies, and so this is basically a sort of a summary of the experimental results, which are expressed by the mechanistic model.
Correct. Yeah.
Thank you. This is helpful. I will maybe explain why at some other occasion when there is more time.
Sure.
So I still don't see any more hands raised. I'm sure people are, they are not tired. They are super.
They are tired.
But so I guess thank you again. Thank you so much for your presentation. Virtual clapping again.